we should be on page 20 talking about the process of meiosis. And at this point, you should know that meiosis is to make sex cells or gametes. And when it makes sex cells, it's going to take a diploid cell and divide it into haploid cells, which have half the amount of chromosomes. And meiosis is going to happen in 2 stages, 1 and 2. So really the only thing on this slide that you need to write down is the purpose, to make sex cells or gametes. The end result of meiosis, you need to know this too, you don't need to write it down just yet, is to make 4 unique haploid cells, which are also known as gametes or sex cells. So the purpose of meiosis 1 is to start with a diploid parent cell and produce 2 haploid daughter cells. So you do need to write this down, but you can kind of simplify it. So you start with a germ cell, is what it's called, and you're going to produce 2 haploid daughter cells by the end of it. Now, don't forget that meiosis is going to start with interphase, and then it's going to do PMAT twice. PMAT for once for meiosis 1 and another time for meiosis 2. But in any cell cycle, we got to start with interphase. The cell is going to grow in size, it's going to prepare its organelles, and it's going to replicate its DNA. And that is key to doing a good job in meiosis, is replicating that DNA first. So remember, meiosis just starts with 1 cell, and it's going to go into prophase 1. And what you need to remember about prophase 1 is this is the preparation phase. And the only thing that you need to write down from this slide is the first bold bullet, that the homologous chromosomes pair up. So remember that homo means same. So these are chromosomes that are the same. So if we are looking at this picture with the peach and the maroon cells, or the chromosomes, each one of these, each X is considered a chromosome. You get 1 from your mom and 1 from your dad. And they are going to pair up, and so there's 4 sister chromatids. So we call that a tetrad. And you might want to circle it at that picture in your notes, that middle picture, and say, that's a tetrad. And what happens with the tetrad during prophase 1 is something very important. This comes up all the time. What's special about prophase 1? Crossing over. Crossing over, not like crossing over to the other side like you're going to heaven, but crossing over of chromosomes, and they're going to share genetic information. So during the crossing over, what happens is they overlap where they're the same genes, and they kind of get tangled up. And during that process, they're going to swap some DNA, and it's going to be for the exact same gene, and this is going to create genetic variation. So the only thing you need to write down under crossing over is the process creates genetic variation or new combinations of genes. If you want to draw yourself a little picture like it has up here, where you show them separate and then crossing over, and that they've swapped genetic information. So teal guy has given him a little bit of his to brown, and brown has given a little bit of hers to teal. And it might be for the gene that codes for your earwax, or how your toenails grow, or whether you sneeze in the sunlight. It's going to be the same gene on each of them that they are going to swap. So then it goes into metaphase 1. And if you remember from PMAT earlier, metaphase middle. So the homologous chromosomes, these pairs of chromosomes, are going to line up in the middle of the cell. And something very special also happens during metaphase 1. It's called independent assortment. And the homologous chromosomes line up just kind of randomly. So in this picture, we have pink and blue. So pink could be on the left and pink could be on the right, or pink could be on the right. In the first one, the first cell on the left over here, the big pink is on the left and little pink is on the left, and big blue is on the right, little blue is on the right. Do you see that? But then in the next cell, big pink is on the left, big blue is on the right, but then it's flip-flopped on the bottom. We have 23 pairs of chromosomes, and they can do as much flipping and flopping, and so you end up with a lot of genetic variation. So both crossing over and independent assortment are really important to give genetic variation to our haploid cells. We want this variation. It means that the offspring are more likely to survive, and that's kind of the whole goal. Think about like oysters. They make thousands upon thousands of embryos at a time, And they're not all going to make it to the right spots, but those that do might have the better variations for cold water or warm water or really fast moving water or really smooth water. And whoever has the most variations are going to be the one that's going to survive. So then we go into anaphase, and just like in mitosis and meiosis, anaphase, they pull apart. Notice they're not pulling apart in the middle yet, though. They're staying in their homologous chromosome pairs and pulling apart as X's. And so the sister chromatids stay together. So if you're going to write this down, just write down that first part, homologous chromosomes, chromosome pairs separate. And if you want to draw yourself a little picture, draw yourself a little picture. Then we're going to go into telophase 1. Telophase 1 also includes cytokinesis, but we never mentioned cytokinesis. Just know that it's there. And what's going to happen is it's going to separate into two cells. So what you want to write down there for telekinesis, or not telekinesis, that's something completely different. Telophase 1 is you have two cells. Now, interestingly, your two cells are still going to have homologous pairs in there. So the end result of meiosis 1, it's two haploid daughter cells. Two haploid daughter cells. And they have a pair of each set of chromosomes, but they're haploid because we count from the middle, from that black dot in the middle of those homologous chromosomes. So then go to the top of page 21, and it says the purpose of meiosis 2. The purpose of meiosis 2 is to form 4 haploid gametes, and they're all going to be unique. They're going to all be their own little individuals. So prophase 2 is the first stage of meiosis 2. Nothing super exciting happens during prophase 2 that you didn't already know. So you can leave that one blank. You can just say it prepares, the centrioles get into place, spindle fibers start attaching, all that good stuff. Metaphase 2, they're going to line up in the middle of the cell. Remember, metaphase middle, and this is going to be very similar to what happened in meiosis. They were X's. Now watch what happens. In anaphase, they're going to pull apart, and they're going to pull apart into their sister chromatids. So that's what I want you to write down. Sister chromatids separate, or the homologous chromosomes separate into sister chromatids, and they're going to move to opposite ends of the cell. And then finally, the cells are going to pull apart into two, and you're going to have 4 unique cells that are haploid. And if you look at that picture with the yellow cells, look at all of those. Each one of those is a little bit different. They're similar, but they're not the same. And that's the point, is to have genetic variation. So the end result is 4 unique daughter cells. And so going over the steps, we start with prophase 1, metaphase 1. So prophase 1, tetrads crossing over, metaphase 1, independent assortment, anaphase 1, pull apart, telophase 1, divide into 2, prophase 2, get prepared again, metaphase 2, they line up in the middle, anaphase 2, the sister chromatids separate, telophase 2, each cell divides into 2, producing 4 cells. So I'm going to draw some cool stuff on the board, and you can kind of model that, but it is like a really intensive thing. So you might want to add to it, but otherwise just kind of keep studying it and making sure you know all the steps of meiosis and what you're starting with and what you're ending with.